Hello and welcome to this new Gen webinar sponsored by Origene, entitled Highlighting the Power of CRISPR AI for Gene Expression Profiling. I'm Jeff Bogaliskis, Technical Editor for Gen, and I'll be your host for today's webinar. Ask any scientist to tell you about CRISPR, and most likely some of the first words out of their mouth would be gene editing tool. And that response would be spot on for the modalities upon which CRISPR cut its teeth. Yet in recent years, investigators have found some even more clever uses for this cutting-edge technology through the regulation of gene expression. Now, the CRISPR activation interference technique, often called CRISPR-A or CRISPR-I, is rapidly becoming an invaluable tool for studying transcriptional regulation and functional genomic screening. Moreover, since many diseases are a function of over or underactive gene expression levels, this sequence-specific approach to repression or activation holds real therapeutic value. Let's meet our presenters for this webinar, who will highlight how a toolbox of modified CRISPR systems is enabling scientists to turn gene expression on using CRISPR-A and off using CRISPR-I. Additionally, we'll learn about some of the latest CRISPR AI tools and reagents and how they are facilitating investigators in their gene editing endeavors. First up is Dr. Luke Gilbert, an assistant professor at the University of California, San Francisco, where his laboratory creates and uses innovative new CRISPR technologies to systematically edit the epigenome and transcriptome of cancer cells, revealing genes that drive cancer progression, metastasis, and drug resistance. Dr. Gilbert will discuss how CRISPR AI functional genomics platform reveals genes that drive complex biological phenotypes. Dr. Mingwan Liu is a product manager at Origene who will tell us about some of the company's latest CRISPR products and how they might benefit your research efforts. But before we begin, I want to remind the audience to stick around for our Q&A session that will happen right after the last talk. Don't be shy. Send in any question you might have for our panelists. It's very easy. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. And we'll try to tackle as many of your questions as we can. All right, I'd like to turn the presentation over to Luke so we can hear about some of the recent work he's been up to. Luke?
Hi, my name is Luke Gilbert. I'm an assistant professor at the University of California at San Francisco. I'm delighted to be talking to you all today. My title for today is Retooling CRISPR to Turn Genes On and Off. So my research is motivated by the idea that within each of us we have at least 200 cell types, each of which has the same genome, but the function of each of these cell types is very different. And this apparent discrepancy between a constant genome and a variable cell function is in many ways explained by the set of genes that are expressed by each cell. And so we know that the genome encodes an instruction book to do really complicated things, and we'd like to know how this book is read out and how the transcripts that are associated with each cell impart function to the cell. And so this is not a profound statement in its own right, but I think in many ways this basic problem has been at the core of many labs' research, but we've lacked good genetic tools to be able to manipulate the genome, the epigenome, and the transcriptome, and therefore be able to decipher how gene function is influenced by the set of genes expressed, or by cell function is influenced by the set of genes expressed by each cell type. So it was with really a great amount of interest that we and many others read Jennifer Dowden and Emmanuel Charpentier's 2012 paper, which described the biochemical repurposing of CRISPR systems as easy-to-use DNA editors. And it's really been, I think, amazing to many people in the community to see how fast these CRISPR systems have exploded into use in biology, where I think there have been papers now showing that you can use them in almost every organism or model organism and many other organisms that are atypical biology research models. And this has really enabled us to increase the pace of research across the biological research community as a whole. At UCSF, we looked at this wonderful system and wondered actually whether there was more that we could use it for. And so in collaboration with initially Stanley Chi, we set out to test whether we could make a nuclease inactive version of Cas9, which we've called dCas9 or dead Cas9. And we've shown over the years that this, initially that this protein still binds to its guide RNA in an appropriate fashion and can bind the right target sequence on the genome. And so this now represents, rather than a site-directed nuclease, an RNA-directed nuclease, represents an RNA-directed DNA binding protein. And so we showed early on in bacteria that you can use this to control transcription by blocking elongating RNA polymerase. In human systems, this doesn't work so well, and so what we've done is appended a series of proteins to dCas9, turning this from a genome editor into an epigenome editor, and this will be a lot of what I talk about today. We've also shown in collaboration with Bo Huang's lab and later Ron Vale's lab that you can use dead Cas9 to image the genome in living human cells, which has additional applications. And so stepping back, I'd like to just describe a couple experiments about how we used dead Cas9 to control transcription by editing the epigenome. And so shown here is one of the very first experiments we did where we fused dCas9 to a scaffold domain called the CRAB domain that recruits CAP1 and HP1 proteins to locally edit the epigenome. And what we were able to show is that we could target this fusion protein to specific sites in human cells and turn off transcription through epigenetic editing. And so below is a RNA-seq experiment where we targeted a GFP reporter and showed that we could turn GFP off or down, and that our ability to control GFP expression was highly specific, such that in this experiment no other genes changed in a significant way. And so we called this method CRISPRi for CRISPR interference, and it's, I think, been useful in a variety of contexts that I'll discuss throughout the talk. But we also wanted to move beyond loss of function genetic perturbations. And so we and others over time have built a series of dCas9 based tools that we can use to activate transcription. And there are version 1, 2, and 3s of these constructs. What's shown here is a version 2 construct that we published a little while ago. And here we've fused dCas9 into a series of epitope domains that can recruit single chain variable fragment antibodies fused to activation domains to activate endogenous gene transcription. And so shown below is an experiment in which we activated the HIV co-receptor, CXCR4, and you can see that version 1 systems didn't work well to activate this endogenous gene, where our second generation systems robustly activate transcription of the CXCR4 gene from the endogenous locus. And so together these now, CRISPRi and CRISPRA, enable us to turn transcription on and off like a light switch, or up and down like a rheostat, and we can program in many experiments how much we want to turn transcription up or down, which is useful for deciphering gene function. These experiments were done in cancer cells or simple cell systems, but our colleagues at UCSF in Bruce Conklin and Martin Kampman's labs have shown that you can use CRISPRi in more complex cell models. And so in these two papers in Cell Stem Cell and Neuron, they demonstrated that you could knock CRISPRi into the genome of iPS cells, and then, as is true in cancer cells, showed that we can control transcription in iPS cells through editing the epigenome in a manner that's efficient, tunable, and reversible. And so we think this has a lot of applications in studying human developmental programs that can be induced from an iPS cell starting point. We think that the use of dCas9 fused to these simple scaffold domains is just a start, though, of our ability to build a toolbox of dCas9 fusion proteins that can edit the epigenome to control transcription or research how epigenetic marks change genome structure or genome organization. And so what we would like to do is be able to at will edit any mark associated with the histone code, as well as DNA methylation. And so over the last couple of years, a series of papers and people have worked on this, including work from Renee Mayer and Rudolf Janisch and Charles Gerschbach, and work from ourselves as well. And we've shown that we can fuse dCas9 to a variety of enzymes that can directly modify the histone code, including histone acetyltransferases and histone demethylases. We can also edit the DNA methylation code by creating dCas9 DNA demethylase and methyltransferase fusions. And so together, these tools, when used on their own or in multiplex fashion, now are a rudimentary toolbox enabling us to edit the epigenome and do discovery biology, or we hope in the future also can be useful for engineering adaptive cellular medicines like CAR T cells. Controlling expression of a single gene, as I've demonstrated on the previous slides, is useful, but we have also built a the ability to control the expression of all of the genes encoded by the genome in pooled experiments, and we call this approach functional genomics. We've shown over the last couple of years that we can clone genome-scale CRISPR-I and CRISPR-A libraries. Starting from DNA oligosynthesis, we clone custom lentiviral libraries. We can turn this from DNA into lentivirus and then infect cells that pre-express Cas9 chimeras for CRISPR-I or CRISPR-A. We then expand cells for a few days and take T0s and then can subject cells to either growth conditions or drugs or toxins or FACS sorting and isolate populations of cells where on the per cell level, knockdown or activation of genes confers some phenotype, and then read these phenotypes out from mixtures of cells using quantitative Illumina sequencing to count the abundance of sgRNAs within different bins or populations of cells, and then infer from the sequencing results how individual genes controls the phenotypes that we're looking at. And so we published a couple of versions of this, and I'll just tell you briefly about one of our early experiments, which is depicted here. So this was the very first loss-of-function CRISPR-I experiment and gain-of-function CRISPR-A experiment that we did. This experiment was done in chronic myeloid leukemia cell line. And what we've plotted here is the genome scale results from the CRISPR-I and CRISPR-A on the same plot. And so CRISPR-I is on the x-axis, CRISPR-A is on the y-axis. Each dot is a gene. And the phenotypes here are log2 normalized per cell division, so they're capped between -1 and 1. What we see in the loss-of-function CRISPR-I results is that we see many hundreds of hit genes emerge. If you were to zoom in and label each of these genes, what you see is you get very strong enrichment for genes that are expected to be commonly essential for life. So these are things like the proteasome, DNA replication, and ribosomal genes. And so even from our first version 1 library, we saw that CRISPR-I does a good job at recovering expected gold standard essential genes. And this gives us confidence that we can control transcription for many of the genes encoded by the genome. In contrast, if we look at the CRISPR-A data, we see fewer hits, but they have a similar magnitude of a phenotype. And when we zoomed in and looked at those hits, what we saw is that a third of these are tumor suppressors, a third of these are transcription factors that control developmental phenotypes, and then there's an enrichment for centrosomal genes. And if we were to take both the CRISPR-I and the CRISPR-A data, we could start to piece together networks of transcription factors that are known to control myeloid development in the stage of development that is where this cancer cell line is trapped, sort of midway through myeloid development. And so what we think this is telling us is that CRISPR-I, or loss-of-function genetic experiments and gain-of-function genetic experiments with CRISPR-A, give you different information about how a cell is wired. And if you want to reveal a full picture of how cells are functioning, loss-of-function CRISPR-I and gain-of-function CRISPR-A together are more informative than either alone. And the last point I'd make on this slide is that with gain-of-function experiments, the level of overexpression doesn't always track with the strength of phenotype. And so this is a gene PLK4 that when we activate it, it gives us a really strong centrosomal duplication phenotype. When we looked at this by qPCR, we saw that we were only getting about a 50% upregulation of this gene. And so unlike loss-of-function experiments where the degree of knockdown or knockout correlates with the phenotypic strength, where we've looked across hundreds of genes, this is not true with CRISPR-A. And so it's just a point worth, I think, making. We've shown over the years that we can do CRISPR-I experiments in a variety of cell types. So this isn't restricted to just easy to work with cell types. And so this slide is a slide from a paper where we were looking at using CRISPR-I to knock down link RNAs using custom CRISPR-I libraries, and we cloned a custom link RNA library for each of the 7 cell types shown here, and then put this into cells and did a simple growth screen, and then looked at the results to identify link RNAs that are required for proliferation. And so whether it's protein coding genes or link RNA genes, which are non-coding genes, we've shown that you can do robust CRISPR-I large-scale screens in cancer cells and normal cells, and the iPS cell data for this link RNA screen is shown below, where you can again see that we get really robust replicate-to-replicate phenotypes that are shown here along the diagonal, and then also that we build in large sets of non-targeting guide RNAs, which lets us estimate where we should call hits. And if you are interested in this approach, there's additional a discussion of this available on the web or on websites. So that all of what I've discussed today so far is relating to controlling the expression of single genes, but we'd really like to be able to control the expression of multiple genes. So you can think of this like a piano, where if you just play one key again and again, it doesn't really tell you what the piano is supposed to do. And so what instead we'd like to do is be able to play all of the keys of this piano at the right intensity, in the right order, and with the right strength. And so this has been difficult to do in human systems, but I think is an aspirational goal where we'd really like to be able to control the expression of 2, 4, or 10 genes at a time to look for how genes work together to enact complex phenotypes. One of the intellectual frameworks that we're working from in thinking about how genes work together to control phenotypes is the concept of epistasis, or genetic interactions. What we know from yeast for a number of years now is that you can make single gene mutations and measure phenotypes, and this gives you information on how single genes that are involved in a given phenotype. But what's more informative is to start to make paired mutations, where you mutate A-B combinations. Most of the time, this will look like some additive effect from the phenotype of mutating either A or B alone, but rarely we'll see A-B mutations that have either less of an expected phenotype, which we call a buffering genetic interaction, and this at its simplest indicates that two genes are in a linear pathway or in a protein complex, such that if you've already disrupted the pathway or complex with either mutation, the combined mutation is less detrimental than expected. And then also rarely we'll see strong synthetic lethal AB mutations, where the phenotype is much stronger than the expected additive phenotype of mutating either A or B alone. And in its simplest interpretation, this usually means that you've found two genes that operate in a parallel biological process, and the cell's capable of dealing with mutation of either A or B, but when you disrupt both of them, the cell's rendered inviable. And so this simple epistasis analysis is useful, but the power of genetic interaction mapping comes from scaling this up to making hundreds of thousands or millions of combination mutations or genetic perturbations. And this then enables you to, at scale, reveal individual genetic interactions that have biological meaning. Medically, we can identify synthetic lethal interactions, which are important for cancer biology, and we can also identify suppressor interactions, which are important for rare Mendelian diseases. Equally importantly, though, when you start to scale up this kind of epistasis or genetic interaction analysis, each gene's genetic interaction profile enables us to cluster individual genes in an unbiased way that reveals gene function. And so we can cluster genes into protein complexes and pathways and organelles, which has been a powerful discovery platform for characterizing unannotated genes in the past. And so all the work until recently had been done in model organisms, mostly in yeast. And so we decided to ask whether we could build a CRISPR-I genetic interaction mapping platform. We stacked the deck in our favor by initially using all validated reagents that we knew were highly active, and then we cloned individual guides into a custom dual sgRNA paired vector library, where we had about 1 million guide pairs targeting 220,000 gene pairs. We made lentivirus out of this DNA library, put it into 2 cancer cell models that pre-expressed CRISPR-I proteins, and then grew the cells for 10 days and harvested genomic DNA, counted the sgRNA abundance by sequencing. This then lets us calculate genetic interactions and cluster genes by their genetic interaction profiles. And in our initial maps, we included genes that were characterized as being part of diverse cellular processes and diverse cellular locations. And we reasoned that with a relatively small map of just around 500 genes, if we could generate clusterable informative information with this diverse of a gene set, then that means that this approach should work for, in theory, larger gene sets or more focused gene sets to construct maps of specific processes or even a map of the whole cell. And so to cut to the punchline, what we did this and held our breath, and what we saw was that we did generate a clusterable map. The map contains a lot of diverse different kinds of biology, but generally genes that function within known areas of biology clustered together. If we were to zoom in on one part of this map, on the mitochondrial subcluster, what we saw was that genes involved in mitochondrial processes cluster together. And then even at the individual complex level shown here, we see that genes in specific protein complexes cluster together as expected. So this is electron transport chain complex 1, which are these NDUF proteins. We hadn't included these on purpose, they just happen to be in the map. What we can see is that all 4 NDUFs in the map cluster together, and then we also see a genome uncharacterized function called TMM261 that clusters just adjacent to these. So this is exactly what we wanted. This is a genome unknown function that is very clearly part of complex 1, and we did a variety of experiments to follow up on this, but is sort of proof of principle that this CRISPRi genetic interaction mapping platform can assign genes to complexes and pathways. We also asked in this experiment whether we could identify unexpected synthetic lethal genes, and this is motivated in part by our desire in cancer therapy to exploit the loss of tumor suppressors. So you can't target a gene that's missing, but as is the case for BRCA mutations and PARP inhibitors, we know that there are ways that we can target cell states induced by tumor suppressor loss for cancer therapy. And so we were interested in our map in druggable genes that were well studied and had unexpected synthetic lethal phenotypes. And I won't walk through all of the data, but we identified this strong synthetic lethal phenotype between FDPS and HUS1 that we were interested in for a number of reasons, including that there was a contradictory phenotype between FDPS and PMBK, shown in this yellow dot, which is an upstream enzyme in this mevalonate biosynthesis pathway. And so we reasoned that this was an interesting biological hypothesis that was complicated, we didn't understand it, but was being nominated by the map. And so we went in and did a series of metabolomic and cell biology and genetic experiments to show that the map had nominated a strong synthetic lethal phenotype between a specific metabolite called IPP that's an intermediate in the mevalonate biosynthetic pathway that we think is the first endogenous allosteric regulator of ribonucleotide reductase, such that if you inhibit ribonucleotide reductase and disable the HUS1, which is part of the 911 DNA repair complex, we get a strong synthetic lethal phenotype. And if you're interested in this, you can read more about it in the paper, but I think it's a good example of a very unexpected complex relationship between 2 well-studied pathways that we never would have looked for that was nominated by the genetic interaction map. We think there will be many more examples of this moving forward. So genetic interaction maps can cluster genes by function and reveal individual hypotheses, but these individual hypotheses, as I pointed out in the last slide, sometimes can take a really long time to follow up on, months or years to figure out what these mean. And so what we're moving towards and what we'd like to do is try to use richer phenotypes to study genetic interactions. And so we've decided to try to do this using single-cell RNA sequencing of genetic interactions. And so I'll walk you through in the last couple slides what we're thinking and where we're headed with this. But we're calling these, rather than genetic interactions, we're calling these genetic interaction manifolds. So this story starts with a realization a couple years ago that several labs had that we can use CRISPR screens combined with single-cell RNA sequencing to generate a really robust merger of 2 amazing technologies that gives us a pooled screen with a very rich transcriptome readout. And so we think that this method of CRISPR screening is really going to be a game changer moving forward. And we're interested in using single gene perturbations in this kind of approach. But what we really wanted to do was ask whether we could do something even more complicated, which is to decipher strong genetic interactions identified in growth-based screens using a transcriptional profiling approach. And so we took a CRISPR-A genetic interaction map that I don't have time to talk about today and mined 287 gene pair and single gene perturbations from this map, and then used Perturb-seq profiling to generate single-cell RNA-seq profiles of each of these single perturbations and double perturbations to learn whether we can use RNA sequencing readouts as a means of understanding strong genetic interactions. And so what we've shown in this paper is that this works and that you can cluster genetic interactions by their transcriptional profiles. We see that we can get very rich readouts where we can distinguish cell cycle arrest or cell death from terminal differentiation. We see that we can distinguish using the transcriptome differentiation towards different cell states such as erythroid or granulocyte or megakaryocyte in this model. These are myeloid cells that are able to dedifferentiate, transdifferentiate, and terminally differentiate. And then we see a variety of other mechanisms within the data as well. And so we think that this will be a very useful transcriptome-based readout platform for understanding developmental biology as well as cell death decisions. And we show in the paper that you're able to decipher additional kinds of genetic interactions that are hard to read out using growth-based genetic interaction profiling. And so in conclusion, I hope I've given you at least a high-level overview of how we view the Cas9 toolbox, which includes both Cas9 and dCas9, and then within the dCas9 branch of this, some of the things that we can do using epigenetic editing and genome imaging. We're focused on expanding these epigenetic editing toolbox towards making multiplexed edits, which we think will be important for controlling transcription in ways that's more faithful to what normal developmental biology does. And then lastly, we're really excited about our new functional genomics tools where we can perturb single genes or pairs of genes, and we can use growth as a readout or fluorescence as a readout or single cell sequencing as a readout, as a very rich readout. And so we think that all these are, should be widely useful, and we're really excited about them. Thank you for your time.
Thanks, Luke. Really excellent presentation. I think we all now have a greater appreciation of the power underlying epigenomic editing. Before we move on to our next presentation, I want to remind the audience once again to stick around for our Q&A session that'll be happening right after this next talk. Send in any question you might have. All you need to do is click the Ask a Question tab on the right-hand side of your screen and hit Submit. It's that easy. Okay, let's get back to the presentations. Ming-Wan, the floor is yours.
Hello, everyone. Thank you for giving me the opportunity to talk about research tools for CRISPR activation and interference. Dr. Gilbert gave you a nice introduction about CRISPR-ANI and its application in the functional screenings. First, I will give you a brief introduction about CRISPR-ANI, then give you more details about the research tools for CRISPR-A and CRISPR-I. So you already know that CRISPR is a simple and efficient RNA-guided genome editing tool. So it has two components, Cas9, the nucleus, and the GRNA. CRISPR technologies has been modified for gene expression modulation. dCas9, that's nucleus deficient Cas9, it can still bind DNA, but cannot cut. So when coupled with the transcriptional activators or repressors, it can regulate gene expression. GRNA bring dCas9 and the transcriptional effectors to the promoter, and then it will activate or repress gene expression. So for effective gene modulation, so where should the GRNA target sequences be located? It should be located around the promoter region. TSS stands for transcription start site. The genomic location uses TSS as a reference. So TSS is 0, and upstream of a transcription start is minus, and the downstream of transcription start is plus. So for minus 200, that means that's upstream of the transcription start, 200 base pairs upstream. For effective GRNA, for gene modulation, scientists analyze the effective GRNAs from their functional screenings. They use a fair amount of GRNAs. So they have observed that for CRISPR-A, if the GRNA is located between -50 to -500, they are more effective for gene activation. However, for CRISPR-I repression, if the GRNA is located between -50 to +500, then they are more effective in repressing the genes. So it's mainly for CRISPRi, GRA should be designed downstream of the transcription start site. It serves as a blockade for transcription. In general, if the GRAs are located close to transcription start site, it tends to be more effective. So here are the CRISPR tools that Origene offers. We have gene-specific knockout kits, every gene, human and mouse. We have many different CRISPR vectors. We also offer custom genome and donor cloning service. Recently, we added the CRISPRa and the CRISPRi products. For AAVS1 ROSA26, that's for transgene insertion into specific location into the genome. We also have functional Cas9 proteins and mRNA. So Origene's CRISPRi vector, we use two repression domains, CRIB and MECP2. So this is all-in-one vector. It has a GRA target sequence cloning site, so GRA will be under U6 promoter and the repression domain dCas9 under CMV promoter. We validated our CRISPRi system by testing human ARPC2 and BRCA1 gene repression. And these are the two GRAs. They are downstream of the transcription start site. So here are the data. For both genes and both GRAs significantly repress gene expression and by over 2-folds. So how can Origene help with your CRISPRi project? One option is you can get this all-in-one CRISPRi vector. You need to do is clone your target sequence in the vector and then transfect into your cells, and your gene will be repressed. If you want to leave the GRN design and cloning work to Origene, we offer custom cloning service. So now I have talked about CRISPRi. Next, I'm going to talk about CRISPRa. CRISPRa is a CRISPR activation, activate gene expression. So it's the transcription activators fused with the dCas9. The commonly used activation domain is VP64, and then GRA brings dCas9 VP64 to the promoter, and then it can activate gene expression. Scientists want to get higher gene activation, so they link several activation domains. There are several models, and the one example is the VPR system. So they linked VP64, P65, and RTA to dCas9. This leads higher gene activation. So next I'm going to talk about CRISPR SAM system. This is the most recently developed and the most robust CRISPR system. So CRISPR SAM stands for Synergistic Activation Mediators. In this system, it has a dCas9 fused with P64 and also need additional activation domain P65 and HSF1. I will give you more details next time. So in this system, dCas9 is fused with the P64. The GRNA is modified. In the loop region, it has MS2 RNA aptamer insertion. So this RNA aptamer binds to MS2 coat protein specifically. So the GRNA brings dCas9 VP64 to the promoter, and then it will activate gene expression. And this system, it has additional activation domains, P65 and HSF1. We call it as enhancer. The additional activation domain are brought to the promoter via interaction of MS2 and the MS2 RNA aptamer in the GRA. So this way, all three different activation domains can activate gene expression synergistically. Origene's CRISPR SAM vector is a two-vector system. The dCas9 AP64 and the GRA will be provided on the all-in-one CRISPR vector. The GRA target sequence can be cloned into the vector. GRA will be under U6 promoter, and dCas9 AP64 under CMV promoter. And the additional activation domains are provided on the enhancer vector. So in summary about CRISPR SAM, it's a very robust CRISPR gene activation system. It's synergistic activation by three activation domains. In Origene's CRISPR SAM, it's a two-vector system. One is the all-in-one CRISPR vector. It contains dCas9 AP64 and the modified GRA. Also, the enhancer vector contains the additional activation domains. So how can Origene help your CRISPR project? You can either get the CRISPR SAM vector kit. It contains the all-in-one CRISPR vector and the enhancer vector. So you can clone your specific GRA target sequence in the all-in-one vector and then co-transfect these two vectors into your cells, and the gene will be robustly activated. To provide convenience to you, Origene offers CRISPR kit that's ready to use gene specific. So every gene we have a CRISPR kit that's transcription activation kit. And you can find the CRISPR kit by gene symbol on our website. So more information about CRISPR kits. So it's a turnkey solution for endogenous gene activation. It's gene specific, every gene for human and mouse. Each kit, it contains 3 GRAs, 1 enhancer, and 1 scramble control GRA. So it's ready to use to activate your gene expression. We validated the CRISPR kit by testing human HBG1 activation. So here are the 3 GRA located upstream of the transcription start site. You co-transfect the GRA and the enhancer into your cells, and scramble serve as a control. The 3 GRAs will be transfected separately, so there will be 4 separate transfections. Inside your cells, the 3 different activation domains will be brought to the promoter and activate your gene expression. Origene also offers several transfection reagents that can help you transfect those vectors efficiently into cells. This is a simple flowchart. And then here is the CRISPR kit, and that's your cells. You prepare for transfection mix and add to your cells. Then for 8 hours, your cells are ready to measure gene expression using qPCR. Here are the data. You can see that all 3 GRAs increase gene expression dramatically, with the GRA1 increased over 2,000 folds. So CRISPR SAM system is a very robust gene activation. We also tested the human ASCL1 activation using the CRISPR kit. So this time we used 2 GRAs. If the cells only transfect with the all-in-one CRISPR vector without the enhancer, and both GRA significantly increase gene expression, about 5-fold. With the enhancer, you can see the gene expression is increased 100 times higher. That's 500 versus 5-fold. So that's why it's called a synergistic activation mediators. In summary, CRISPR and AI are simple and efficient gene modulation tools. And all splice variants, if they are transcribed from the same promoter, will be affected since the CRISPR and AI will activate on the promoter. So it will affect all splice variants of that gene. Origine's CRISPR vector employs 2 repression domains, and the CRISPR SAM system can upregulate gene expression by over 1,000 folds. We also have genome-wide CRISPR kits that's available for you, ready to use, and you can find the CRISPR kit by gene symbol on our website. Please visit us to find more information on our CRISPR and CRISPRi website. If you have more questions, please contact our technical support team. Thank you for your attention.
Very nice presentation, Ming-Wan. Thank you. Some really great products that Origene has for CRISPR researchers. All right, the Q&A is about to begin very soon, and I want to remind the audience once again to send in their questions for our panelists so we can have a very lively discussion. We'll try to get to as many of your questions as we can. Again, simply type your question into the Ask a Question tab on the right-hand side of your screen and then hit submit. So it looks like we have a bunch of really great questions that have come in already. So I want to thank all the audience members that have sent in questions. But for anyone still sitting there deciding whether they should send in a question or not, please do so. We'll try to have a very lively discussion and get to as many of your questions as possible. Just bear with us for a moment while we get the logistics squared away, and we'll begin the Q&A presently.
All right, everyone. We've got a bunch of really great questions that have come in, so thank you to the audience for sending those in. Let's get to them. Our first question is going to be for Luke. Luke, one of our audience members would like to know, how did you connect the transcription factor to the dCas9?
Yeah, thanks, Jeff. This is a surprisingly important part of CRISPRi especially, and also CRISPRA. We know some of these domains that edit the epigenome are unstructured, and we need the right linker to connect dCas9 to these domains in order to maximize epigenetic editing. And so you can get more details off of our website or from Adgene, but this is a really crucial part. Our current preferred linkers are sort of long and unstructured, but we also want to avoid proteolysis within the linker. So there are a number of commercially available CRISPRi constructs that don't work well, and I would be careful about this.
Great. Thanks very much, Luke. Mingjun, a question for you. For your CRISPR activation kit, can I make stable cells?
That's Jeff. Yes. The CRISPR kit, in the all-in-one CRISPR vector, it does not have a momentum selection marker, but the enhancer vector has a neomycin selection marker. So people can select with neomycin resistant to make stable cells.
All right, thank you very much. Luke, back to you. What is the efficiency of the expression activation? In my usage of the SAM system, I reached 30% of the surface expression.
Yeah, so there's two parts to this question. One is whether all of the cells within a population are expressing the CRISPR protein. That can be tricky to achieve in certain models, and then sometimes we will go to clones that have stable protein expression across a population of cells. But for other genes, you can have heterogeneity in activation. This is an active field of research and looking into where CRISPR-A has heterogeneity and where you get a homogeneous expression across the population of cells, and seems to depend on the endogenous locus. But I think more relevantly to this question, I don't think that fold activation is a good measurement for CRISPR-A. We've shown in this 2019 Science paper that if you activate a gene that's not expressed, essentially you're dividing by 0, and so you can get near infinite fold change expressions. And so I think FPKM or TPM or other RNA-seq measurements where you measure the absolute transcript abundance is a fairer way to ask how much you're activating genes that are not expressed, lowly expressed, medium, or highly expressed. And for highly expressed genes, we've shown that modest increases in gene expression can have really profound effects on cell physiology.
All right, thank you, Luke. Mingjun, question for you. An audience member asked that if they need to do a gain-of-function study, should I choose CRISPR-A system or a cDNA clone?
That's a good question. Yes, for CRISPR-A, the pro for CRISPR-A is very straightforward. You just co-transfect the CRISPR-A vector with the enhancer in our system. You can activate gene expression. The number 2 for the advantage that since you are activating from the endogenous promoter, so it affects all the splicing variants that started from that promoter. So that's you will get a full function of the gene. So the cons, disadvantage of the CRISPR-A, for gene modulation using CRISPR-A is sometimes the effect depends on the genomic context. So sometimes just the CRISPR-Cas9 and the transcription factors cannot access to the DNA. Sometimes the DNA is nucleosome occupied or in the heterochromatin. And the transcription regulation is a constant occupance of the transcription factor Cas9. So that's the disadvantage of the using CRISPR-A. For cDNA clones, so far it's still most common, most direct form of doing gain-of-function study. Also the second advantage for cDNA clone is the cost. Now they are very economic. A lot of the cDNA clones are less than $100. As a matter of fact, Origene has over 20,000 of them are priced less than 100. So, but for cDNA clones, sometimes the size is very big, it's huge. It may take long time to make it. So then you can use CRISPR-A and try CRISPR-A to see you can do gain of function for those larger size of genes. I think that's all.
Great. Thank you very much, Mingjun. Luke, another question for you. An audience member would like to know if you looked into the specifics of the isoforms in the CRISPR-A system.
Yeah, so I'm going to answer this question in 3 ways. One was already addressed in that CRISPR-A is useful for activating large genes that can't be easily packaged into lentiviral systems. But this isoform question is a second advantage of CRISPR-A, where usually when people use a cDNA for an experiment, you have to make an assumption about which isoform you want to express, and people rarely test all isoforms expressed within a cell. So with CRISPR-A, we don't have control over isoform control of expression. The cell still chooses which isoforms to express, but you sample naturally different isoforms that can be expressed. And so we would love to be able to build a CRISPR tool that selectively edits different splice isoforms, but for now, we just have to let the cells choose. And the last thing about the comparison of CRISPR-A to cDNAs is that I think CRISPR-A really starts to shine where you move away from controlling single genes towards pooled functional and genomic screens, where cDNA libraries have been quite difficult to use in pooled fashion. And so CRISPR-A, where you just need to express small guides, is quite easy for activation of large sets of genes.
All right, great. Thank you, Luke. Mingjun, a question for you. What version of the CRISPR SAM system does Origene have? Is it version 1, 2, or 3?
Okay. For CRISPR-A and SAM, I think the version 1, 2 in overall during CRISPR technology development. So for CRISPR-A SAM, there's only one version that's published by Dr. Feng Zhang's group. So, and it's a very robust activation system. So far, I believe that's one of the most robust system for CRISPR-A.
All right, thank you. Luke, question for you. Using the CRISPR-A, CRISPR-I system, the expression of dCas9 protein is very low, as analyzed by Western blot. How do you improve the expression of dCas9?
Thanks, Jeff. Actually, can I comment on the last question as well? So there's a paper in Nature Methods from George Church's lab comparing SAM to Viper to Suntag, and you can refer audience members to that if you want to get a direct head-to-head comparison of the systems. They seem to vary depending on the gene you're targeting. So as to the second question about the level of dCas9 protein, this is a complicated question, and it's a good question. It seems in our analysis that the expression of dCas9 is usually not correlated with it being highly functional. So another way to say that is we don't need very much expression of dCas9. Often the amount we're expressing is very low, like on the order of 1,000 proteins per cell if you were to measure absolute copy number. And despite this, dCas9 is a really efficient enzyme that's able to search across the genome and bind to its target sequence at really high occupancy, even at low protein expression. And once you've achieved sort of maximal target occupancy of dCas9, any overexpression beyond that just induces off-target effects. And so we don't worry if we have low expression of dCas9. We put dCas9 in with lentivirus, usually sort twice, to achieve a pure population of cells that express dCas9. And you do need protein expression, obviously, but we, like in a variety of experiments where we've selected for higher and higher dCas9, you don't see increased activity. And so I would caution audience members to define your function, define your systems functionally using either target gene knockdown or activation, or even better, we like to express guide RNAs targeting essential genes and look at cells dropping out of the population over time. And there's additional commentary on this on the web if you want a series of positive controls for these kind of functional experiments.
Thank you, Luke. Mingjun, I think this question is going to be targeted to you. An audience member would like to know that they ask if they want to regulate expression of protein of interest and wish to use CRISPRi instead of inducible shRNAs. Maybe you could comment on that.
Sure. With the CRISPRi, in our vector is all-in-one. That's a fairly easy. The dCas9, CRAB, and the MECT2 is already expressing the vector. Also, there's a gRNA targeting cloning site in the vector too. The researchers just need to clone the gRNA target sequence in the vector and then just transfect the vector into their cells. They should be able to study if it can repress their gene expression. And also, Origene offers custom cloning too. So if they want Origene to design the gRNA and a clone into the vector.
All right, thank you. And it looks like we have time for one last question for Luke. Luke, can you regulate the expression of CRISPR-A, i.e. gene expression of the target gene, control the expression up or down like a dial or on of a light versus like on-off?
Yeah, thanks, Jeff. So for on-off, you just want to pick the maximally active guide RNA for either CRISPR-I or A. For turning gene expression down or up, we've referred to this sometimes as a pseudoallelic series. And there's two ways to do this. One is we know you can move where the transcription factors bind relative to the transcription start site to tune activity. So farther away, you get weaker activity, and closer into the maximal window of activity, you get stronger activity. But a second, maybe more elegant way has been nominated recently by Jonathan Weissman's lab in a paper on bioRxiv, where they've intentionally included mismatches in the sgRNA that can programmably decrease or increase transcription. So if you're curious about that, take a look on bioRxiv.
All righty, thank you, Luke. And with that, we've come to the end of our webinar. So I'd like to remind everyone the webinar will be archived on our website at www.genengnews.com for up to a year. So if you miss any parts of it, you can watch it again, or feel free to forward the link to any of your friends or colleagues. I'd like to thank Luke and Mingjun again for their very informative presentations, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Origene for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.